This year, researcher Rich Barclay from the Museum of Natural History started working out at CERC with some of our citizen scientists. Rich is a paleobotanist, which means he studies plants that existed millions of years ago. We are interested in learning what our atmosphere was like millions of years ago. Our planet's atmosphere is composed of many gases, one of which is carbon dioxide. We know that plants, including ginkgo, survive by photosynthesis, that is, converting energy from light and carbon dioxide into sugars. The ginkgo tree was cultivated early in human history and has been found in fossils that are over 200 million years old. By looking at fossilized ginkgo leaves and modern ginkgo leaves under a microscope, we are able to see the stomata of leaves. Leaves breathe by opening tiny pores called stomates under their leaves, which take in carbon dioxide. However, when the stomates are open and the plant is breathing, the plant also loses water. Researchers have found that there is an optimal number of stomata on each leaf that allows a plant to be most effective to ensure that they don't lose too much water but also get enough carbon dioxide. At CERC, Rich and other citizen scientists want to learn about the future. We have set up a series of chambers with ginkgo trees and have been pumping different levels of carbon dioxide into those chambers to see how ginkgo trees and their stomates will react in the future.
It's fantastic to have volunteers involved. There's no way that we can do this project without the help of volunteers. The project requires daily maintenance, where we have 15 trees and 4 different atmospheric treatments. Ambient out here is about 400 parts per million for CO2, and then we have plants at 600, 800, and 1,000. Maintaining the horticultural aspects, keeping the trees alive in these difficult conditions, and then maintaining the CO2 values to close to targets. We have to adjust the CO2 a couple of times a day. Sometimes things drift off for one reason or another, and they've been helping with all aspects of that. The volunteers that have been out there have been tagging individual positions on the plant, so we know exactly where those leaves come from. And then we'll resample that same location every year, because it's a multi-year project that'll probably take 5 years to be fully complete. So initial data that is equivocal, yet it's going to take years for the plants to adjust to those conditions. So essentially, as CO2 rises, you get a decrease in the number of pores, and we're generally seeing that happen, but the data we've been able to collect really only reflects about 6 months of exposure, and it's going to take a couple of years for those plants to fully acclimatize to being in, pretty difficult conditions.
This is the first year of the project, so we're still waiting for the results, but pretty soon we'll be counting the stomata on leaves collected from the living trees also.
We thank you for all the time. There's been something on there. I can't keep track at this stage of how many people have been involved, but each one of the people that have been involved have really done a bang-up job.